Imagine an adversary that, well, evolves faster than we can. A master of disguise, constantly outmaneuvering our body's best defenses.
That really is cancer, isn't it? Cunning, adaptable,
Mm.
incredibly complex.
Exactly. And today we're really going to dive deep into the, let's say, the bewildering world of cancer biology. We want to explore those sophisticated strategies cancer cells use to grow, to spread, and crucially, to hide from our immune system.
It's like looking at cellular warfare in action, how these rogue cells just get so good at navigating and, well, exploiting our own biology.
It is. So for this deep dive, we've pulled together a lot of information, key biological explanations, some really compelling real-world case studies.
Yeah, the vignettes really help illustrate the mechanisms.
They do. Our mission here is to unpack all these complex mechanisms, turn the intricate details into something clear, memorable, basically give you a shortcut to understanding the science. Hopefully you'll have some aha moments.
Definitely. Let's get into it.
Okay, let's start with spreading. A cancer cell starts in one place, right? So how does it suddenly become this traveler inside the body? I mean, it's not like they sprout legs or anything.
No, not quite legs, but the journey, it starts with something that sounds simple. Just breaking free from the main tumor. But it's not random at all. It's a highly orchestrated process, like a blueprint for invasion.
Okay, so step one is breaking free, detaching. How? Normal cells seem pretty stuck together.
They are. Think of normal cells like bricks in a wall held together by mortar. A really important part of that mortar is a protein called E-cadherin. It handles cell-to-cell adhesion.
Right.
Now, cancer cells, they figure out how to turn down the production of this E-cadherin. Less E-cadherin means weaker mortar. The connections loosen, and individual cells can kind of wriggle free.
Dissolving the mortar. Okay, so they're loose. What next? They can't just float around forever, presumably.
No, exactly. Once they're detached, they need something new to grab onto. And that's where the extracellular matrix, or ECM, comes in, especially proteins in the ECM, like laminin and fibronectin.
Like sticky landing pads.
Pretty much, yeah. And cancer cells have these special receptors on their surface. They're called integrins. These integrins are designed specifically to bind to laminin and fibronectin.
Oh.
We actually saw a case, a 58-year-old man with lung cancer. His malignant cells showed increased expression of these integrins, the ones that bind laminin and fibronectin.
And what did that finding tell the doctors?
It's a big clue. It signifies enhanced tumor invasion through the basement membrane. It shows the tumor is actively gearing up to move, to spread. It's a really critical step for metastasis.
Okay, but just attaching isn't enough, right? They need to actually move through tissue.
Yeah.
How do they clear a path?
Right, and this is really quite clever or sinister, depending how you look at it. They don't just attach, they actively clear the way. They pump out this cocktail of enzymes.
Enzymes? Like what?
Things like metalloproteinases, MMPs, and collagenases. Think of them as molecular bulldozers. These enzymes break down the stuff in their way, like collagen IV in the basement membrane. They literally dissolve the barriers.
Wow. So detach, attach, degrade, and then they move.
That's step 4, migration and invasion. Path cleared. They start moving through that degraded ECM. Often they're guided by chemical signals, cytokines, and they can spread through the blood, the lymphatics, or sometimes just by seeding directly into body cavities.
This whole 4-step process, detach, attach, degrade, migrate, it's not just fascinating biology, it's like a tactical map for researchers, right? Each step is a potential target.
Absolutely. Understanding how they spread is fundamental to stopping them.
Okay, here's something else that's always baffled me. Most of our cells have a limited lifespan, right? Like it's a built-in expiration date. They divide, they age, they stop. How does cancer just ignore that, become immortal?
Yeah, that's one of the core problems. So normal cells, our somatic cells, have these protective caps on the ends of their chromosomes. They're called telomeres.
Right, I've heard of those.
And every time a cell divides, these telomeres get a little bit shorter. It's like a countdown timer. Eventually, they get too short, and the cell gets the signal to stop dividing. It enters senescence, basically cellular old age. It's a safety mechanism against runaway growth.
But cancer cells naturally find a loophole.
They do. They find a way to reactivate an enzyme called telomerase. Normally, this enzyme isn't very active in most adult cells, but telomerase is a reverse transcriptase, and its job is to add these repetitive DNA sequences. TTGG repeats back onto the ends of the chromosomes.
That's who rebuilds the telomeres.
Exactly. By reactivating telomerase, cancer cells prevent their telomeres from shortening with each division. They bypass senescence. They effectively become immortalized, capable of dividing endlessly.
They just keep resetting the clock. We saw a perfect example, didn't we? A 64-year-old man, lung cancer, high mitotic activity. And the key finding was upregulated telomerase.
Yes, and the explanation was clear. Maintenance of chromosomal length by adding T-G repeats. That vignette nails it. It's how cancer cells write their own rules for immortality.
Which makes telomerase a huge target, I imagine.
A massive target. If we could selectively shut down telomerase just in cancer cells, we might be able to force them to age and die like normal cells.
Okay, so they can spread, they can live forever. But why do they end up in specific places? You hear about lung cancer going to the brain or prostate cancer to the bone. It doesn't seem random.
It's definitely not random. There's a term for it, organotropism. It's the tendency for metastatic tumors to prefer specific organs.
Organotropism, right.
And the classic explanation is the seed and soil hypothesis. The idea is that the tumor cell, the seed, can only really thrive and grow if it lands in a compatible microenvironment, the soil of a particular organ.
So the destination organ has to be receptive somehow. What makes it receptive?
It's down to molecular interactions, specific adhesion molecules, things like integrins again, and selectins, plus chemokine signals between the tumor cell and the cells lining the blood vessels in the target organ. It's like a molecular handshake or a homing signal.
A homing signal guiding them. Can you give us some common examples? Where do specific cancers tend to set up shop?
Sure. Lung cancer is a good one. There's a mnemonic doctors use. Lung BBLA. It stands for brain, bone, liver, and adrenals. Those are the frequent spots.
BBLA.
And we saw case examples, didn't we? Lung adenocarcinoma showing up as multiple ring-enhancing brain lesions, or squamous cell lung cancer presenting with a large unilateral adrenal mass. Very typical patterns.
What about others?
Prostate cancer is well known for going to bone. It often causes osteoblastic lesions, meaning it actually stimulates new bone growth there. Colon cancer, because of where its blood drains, very commonly goes to the liver first.
Liver first from the colon, right?
And breast cancer. Well, breast cancer is notorious for being able to go almost anywhere. Bone, lung, liver, brain are all common sites. You could maybe remember that as BBLL.
BBLL for breast. So for you listening, understanding this organotropism isn't just academic. It helps explain why patients might have certain symptoms, and it tells doctors where they need to look if they suspect the cancer has spread. It's knowing the enemy's likely escape routes.
Precisely.
Now, this leads to another big question. Our immune system is incredible, right? It's constantly patrolling, looking for abnormal cells. So why doesn't it just wipe out cancer cells as soon as they appear? How do they slip past?
That's maybe one of the most fascinating and frustrating parts of cancer biology. They develop these really ingenious ways to evade immune surveillance. It's like they have a whole toolkit for becoming invisible or disarming the guards.
Okay, what's tool number one? How do they hide?
A major strategy is messing with MHC class I molecules. Normally, our cytotoxic T cells, the CD8 era T cells, recognize little bits of tumor protein antigens presented on these MHC class I molecules on the cell surface. Recognition leads to killing the tumor cell.
So MHCI is like the cell's ID badge, showing what's inside.
Exactly. And what tumor cells do is they downregulate MHCI. They basically stop showing their ID badge.
So the T-cell patrol just passes by, no ID, can't check you.
Pretty much. No ID badge, MHCI, security, CDA, can't stop intruder, tumor. It makes them invisible to that main arm of the adaptive immune system. We saw that case, the 58-year-old man with squamous cell carcinoma. His tumor had markedly decreased expression of MHC class I molecules. That helps it survive.
But hang on, I thought if cells lose their MHCI, another type of immune cell, the NK cells, natural killer cells, are supposed to spot that and kill them.
You're absolutely right. It is a paradox. NK cells are specifically looking for cells that lack that self-signal of MHCI. So hiding from T cells should make them sitting ducks for NK cells.
How do they get around that?
Well, cancer has countermeasures for the countermeasures. We'll get to NK cell evasion in a moment. But first, it's not just about hiding. They also actively slam the brakes on the T cells that do manage to see them.
Okay, how do they hit the brakes?
They use another pathway. Many tumor cells increase their expression of a protein called PD-L1. This PD-L1 on the tumor cell binds to a receptor called PD-1 on the T cell surface.
PD-L1 on tumor, PD-1 on T cell.
Right. And that binding, PD-L1 to PD-1, sends an off signal to the T cell. It inhibits T cell activation. It leads to what we call T cell exhaustion. Basically, it neutralizes the attack.
So hiding, hitting the brakes.
Well,
what else?
They create a hostile environment for immune cells. They actively secrete molecules called immunosuppressive cytokines. TGF-β and IL-10 are key examples.
Then what do those do?
These cytokines directly suppress the activity of T cells and NK cells, but they also do something sneakier. They attract regulatory T cells, or TREGs, into the tumor. TREGs are immune cells whose natural job is to dampen immune responses to prevent autoimmunity. So the tumor recruits the immune system's own regulators to protect itself.
Wow. Recruiting the peacekeepers to protect the criminals.
In a way, yes. We had that lung cancer example where the tumor cells were secreting TGF-β, and the result was recruitment of regulatory T cells, TREGs, leading to suppression of anti-tumor immune responses. It creates this immunosuppressive bubble.
Okay, I'm still thinking about those NK cells. If MHCI is low, NK cells should attack. How does cancer stop them?
Right, the advanced trick. So NK cells don't just look for missing MHCI, they also look for stress signals on cell surfaces. These are molecules like mica and MICD, which get turned on when cells are stressed or cancerous. NK cells have a receptor, NKG2D, that recognizes these stress ligands.
The MICD are like alarm bells for NK cells.
Oh, exactly. But here's the trick. Some tumors can actually shed soluble versions of mica and MICB into the bloodstream.
Shed them? Like decoys.
Precisely. These soluble MICAB proteins float around and bind to the NKG2D receptors on the NK cells before the NK cells even reach the tumor. It jams their targeting system.
So the NK cells are effectively blinded before they get there. We saw that specific case, didn't we? Non-small cell lung cancer, low MHCI, and secreting soluble mica.
Yes. And the mechanism was inhibition of NK cell activation by blocking NKG2D receptors. So that tumor had figured out how to evade both the CD8 plus T cells by losing MHCI and the NK cells by shedding the decoys. A double whammy.
It's just layers upon layers of evasion, this constant cat and mouse game. It's incredible.
It really highlights the challenge we face.
But knowing all this, knowing how cancer plays defense,
Yeah.
surely we can use that knowledge offensively, turn the immune system back against the tumor.
And that is exactly what modern immunotherapy aims to do. It's been a revolution in cancer treatment. Instead of just poisoning the cancer, we're empowering or redirecting the patient's own immune system to do the job.
Okay, so let's talk about releasing those brakes we mentioned earlier, the PD-1, PD-L1 pathway.
Right. Immune checkpoint inhibitors are designed to block those braking signals. You have drugs that block CTLA-4, which is another early brake on T-cell activation. Ipilimumab is an example.
Ipilimumab, right?
And then you have drugs that block the PD-1 receptor on the T-cell itself, like nivolumab or pembrolizumab, or drugs that block the PD-L1 ligand on the tumor cell, like atezolizumab. By blocking either side of that interaction, you prevent the off signal and allow the T-cells to stay active and attack the tumor.
Taking the brakes off. But unleashing the immune system like that can have side effects, can't it?
Absolutely. Because you're removing a natural control mechanism, the immune system can sometimes attack healthy tissues too. We saw examples like severe diarrhea and colitis with ipilimumab or immune-related pneumonitis lung inflammation with nivolumab. Managing these immune-related adverse events is a key part of using these therapies.
Okay, so checkpoint inhibitors release the brakes. What about making the immune cells better fighters, those personalized warriors?
You mean CAR T-cell therapy? Chimeric antigen receptor T-cell therapy. This is incredibly sophisticated.
How does it work, basically?
You take T-cells from the patient's own blood. Then, in the lab, you genetically engineer them to express a specific receptor, the CR, on their surface. This CR is designed to recognize a particular target on the patient's cancer cells, like the CD19 protein on B-cell leukemias and lymphomas.
So you're giving the T cells super specific targeting instructions.
Exactly. Then you grow millions of these engineered CAR T cells and infuse them back into the patient. They become a living drug, hunting down and killing cells with that target protein.
Amazing. But potentially risky, too. We had that case of the teenager with B-cell ALL who got CAR T.
Aaron developed fever, low blood pressure, high IL-6. Yes, that's cytokine release syndrome, or CRS. It happens because when the CAR T cells get activated and start killing tumor cells, they release a flood of cytokines, causing widespread inflammation. It can be serious, but we now have ways to manage it, often with drugs like tocilizumab that block the IL-6 signal.
So checkpoint inhibitors, CAR T cells.
Yeah.
What about simpler targeted approaches like antibodies?
Monoclonal antibodies are another major pillar. These are lab-made antibodies designed to stick to specific proteins on cancer cells. Trastuzumab, for example, targets the HER2 protein in some breast and gastric cancers. Rituximab targets SEED2-mab, blocking HER2 signaling. Others, like rituximab, act more like flags, marking the cancer cell for destruction by other parts of the immune system, like NK cells or complement. Though they can also have side effects, like potential heart issues with trastuzumab.
And finally, Can we use the immune system for prevention? Vaccines.
Yes, definitely. The HPV vaccine is the prime example. It targets the L1 capsid protein of the human papillomavirus. By generating immunity against the virus, it prevents the infections that cause most cervical cancers and several other cancer types too. It's true prevention through immunity.
So for everyone listening, these immunotherapies, they really feel like a turning point, don't they? We're learning to use the body's own incredibly powerful defense system.
It's a fundamental shift. We've gone from fairly blunt instruments to highly targeted, biologically sophisticated strategies. It's hugely promising.
So we've covered a lot today. We've journeyed through how cancer spreads using that invasion blueprint,
How it achieves a kind of immortality using telomerase.
how it specifically targets certain organs, organotropism, the seed and soil idea.
And those really complex, multi-layered ways it hides from or disarms our immune system, downregulating MHCI, using PD-L1, secreting immunosuppressants, even shedding decoys for NK cells.
And then finally, how we're fighting back. Taking the brakes off with checkpoint inhibitors, creating super soldiers with CAR-T cells, using targeted antibodies, and even preventing cancer with vaccines.
It underscores that cancer isn't just one thing. It's this incredibly complex, evolving adversary. And research is constantly peeling back new layers, finding new vulnerabilities.
It really is a continuous battle.
Yeah.
Which brings us to a final thought. Considering just how adaptable cancer is, all these layers of immune evasion we've talked about, what new tricks, what new rules might cancer cells be developing right now? Things we haven't even discovered yet.
That's the question that keeps researchers up at night, isn't it? And it's what drives the search for the next breakthrough. It's a constant race between the disease's evolution and our understanding.